Hi students. Today's video lecture is going to be on the third type of biomolecule that we're learning, proteins. So we've talked about carbohydrates, we've talked about lipids, and now we're going to start discussing proteins. So out of all the biomolecules, proteins really have the most diverse function in our body. Anything that's doing anything in our body is a protein. So proteins are the enzymes that facilitate chemical reactions. Proteins are the antibodies in our immune system that help fight diseases. Proteins are the molecules on our cell membrane that allow things to enter and exit the cell. Proteins are what attach to our skeletal parts and allow us to move. And the key as to why they have such a diverse function in our body really lies in their structure. So the structure of a protein allows there to be such a diversity in forms, and all these different forms allow different functions. So as we've talked about other biomolecules, we're going to learn what proteins are made up of, the monomer, and then those building blocks join to make the polymer. So the building block or the monomer of a protein is called an amino acid. I'm sure you've heard about this word before, so we're going to discuss the structure of amino acids. So different amino acids, the monomer, will bond together, form covalent bonds. The actual name for the covalent bond between amino acids is a peptide bond, but it's just a type of covalent bond. And when you have a lot of amino acids bonded together, it forms a many peptide because those are peptide bonds. So the polypeptide is the polymer made up of repeating monomers, the amino acid. Once a polypeptide is formed, which is basically just the chain of amino acids, that chain is going to start to bend and fold. And that bending and folding is going to vary between the different proteins. And we're going to talk about why. And once that polypeptide is bent into its final structure, then we refer to it as a protein. So we start with the amino acids. The amino acids bond together to form the polypeptide, the chain of amino acids. And then that polypeptide chain bends and folds into the final protein that can perform a specific job. Here's the table that we've looked at before, which talks about all the different biomolecules on the left. We've talked about carbohydrates, lipids, now we're on proteins. The subunit or the monomer, the building block, is an amino acids. The polymer, the larger molecule, is a polypeptide. All right, so now a little bit more about the building block or the monomers, which are these amino acids. So there's 20 different amino acids, and each amino acid has a few things in common, which is why they're called amino acids, but they vary in one region. So if you look at this generic structure of amino acid, you see that on the left is what that we refer to as an amino group, which is the N, the H2N, 2 atoms of hydrogen, 1 of nitrogen. That's bonded to, this line represents just a covalent bond, to a central carbon that has a hydrogen coming out of it, and that central carbon is bonded to this other group, right, this acidic, which is a carboxyl group, COOH. That's always there in any amino acid. What differs between the 20 amino acids is what is coming out of the central carbon, this R group. So this R group, in this image, it's just showing you a generic R. But there are 20 unique types of amino acids, and each one of them has a different R group. Okay? So we're going to talk a little bit about the different R groups and how it gives each amino acid certain properties. I like to kind of make the analogy of words in the English language. Like we have 26 different letters in the alphabet, and with those letters we can combine them in different orders to make almost infinite number of words. That's the same idea with a protein. We have 20 different amino acids, and we can join them in different combinations, and those different combinations will cause each polypeptide to bend in a different way to make infinite numbers of different types of proteins. All right, so here, this is just showing you some of the different amino acids. All right, and if you look at this picture, let's look at the top, okay? Sample amino acids with non-polar R groups. So if you look at all of these 5 different amino acids, the part that is not shaded is the part that is common to all amino acids. You see that H2N, the central carbon, the other carbon with the O and the OH. This is a little different. It's showing you in the ionized form, but that's basically referred to as a backbone. What is highlighted is the R group. So the R group, remember, is what varies between the different amino acids. So the 5 here under this orange box and that have the R groups highlighted in orange are all non-polar. So these amino acids won't interact with water. So that will cause, if they are linked in the chain, for them to respond in a certain manner. It will cause the polypeptide to bend. It will cause any amino acids that are these to go away from water. Below that, you see another group of amino acids that are polar. So these will interact with water. So you can see that if you have a chain that's polar and a chain that's non-polar, it's going to cause it, if it's surrounded by water, for it to bend in a different way. All right, and then here on the bottom, there's other ones which actually have ionized R groups. So now these are the different amino acids. We join them together, right? Just like we joined all the monomers together. We join them, we remove certain atoms, the OH of this amino acid, the H of this. All right, that's actually referred to as a dehydration reaction. We release water, and now see peptide bond, which is just a covalent bond. And now we have a chain with 2 amino acids linked together. If we keep doing this, we're going to make a bigger and bigger chain, and we're going to have a polypeptide. The key into how proteins function is their shape, right? It's their shape. So for example, the shape of a key is really important because it will open certain locks and not others. That same idea can be held true with proteins. Their shape is critical to what they do in our bodies. And so we're not going to get into too much detail here. I just kind of want you to have the idea of this. But we talk about shape and how a polypeptide bends in terms of levels, right, of structure. Primary is like the chain of the amino acids. Then secondary is how that chain always folds upon itself. And tertiary is how then that slightly folded chain folds again. And quaternary refers to if we actually have 2 polypeptides folded, sometimes they'll join to make a really mega large protein. This is what I just said in a little more detail. So primary structure is the linear sequence of amino acids, right? Just the chain, right? Hundreds of thousands of different polypeptides can be built from just the 20 amino acids because each polypeptide can have a different number of amino acids and in a different order. Changing the sequence of the amino acids can produce different proteins. Secondary structure is always what forms between those constant regions of the amino acids, the ones that are the same, and those different shapes are referred to as alpha helices or pleated sheath, right? And then tertiary is the overall, when the secondary folds again into a 3-dimensional polypeptide. And quaternary is more than one polypeptide folded together. So I think this picture will help. So if we start on top, see where it says primary structure. Primary structure is just the chain of amino acids. And here, each amino acid is represented as one bead on the chain. So this is a whole chain of amino acids, right? And that's the primary structure. The secondary structure causes these chains to make what's on the left either an alpha helix, this helical shape, and that's just by interaction of the constant regions of the amino acids, or this kind of more accordion shape on the right, which is a pleated sheet. And those secondary shapes, the alpha helix and the pleated sheath, will fold upon themselves because of the characteristics of those R groups to form this final tertiary structure, which is here where I'm highlighting. So this is one polypeptide that has folded into its tertiary structure, all right? This maybe could be a protein, but sometimes more than one of these come together to give quaternary structure, right? So in a quaternary structure, you would have two or more folded polypeptides put together. And there's a video about this that I've also posted from the textbook that helps you understand this. The one thing about proteins, I'm going to give you an example that you've probably seen before, is that proteins, because their shape is so specific and it's held together by these bonds and interactions between the R groups, is that what can happen to them is that under certain harsh conditions, that shape can be compromised. It can fall apart. So for example, have you ever fried an egg on the stove, on a pan? All right, when you open that egg up and you put it on the frying pan, you have the yolk and then the egg white. And initially that egg white is transparent. You can almost see through it, right? That's almost all protein, okay? That's almost all protein, that egg white. And when you put it on that hot pan, that's a really harsh condition. And all of a sudden, that egg white turns from transparent, translucent, to white. That's denaturing. That protein that made up the egg white was put under high temperatures, and it caused all of the shape to change, which made it appear different to our eyes. That's also the danger in having a really high fever. If you have a really high fever, the enzymes that allow all the reactions to happen in your body will start to denature, and then they can't do their job anymore, because the job of a protein, it's based on its structure. And if we start to change the shape, they can't do their job anymore. So exposure of proteins to chemicals also, or very acidic changes in pH, if you put in an acid, or high temperatures can disrupt their structure, and that's referred to as denaturing. All right, that wraps up proteins. Take care, everyone.